Each of us will speak. Andy's going to talk first about the big paper that came out in PNAS. I'll talk a little bit about classification. Matt will talk about the things that we did to develop the new classification, and I'll just come in at the end to wrap things up. So, Dr. Alverson, the floor is yours.
All right, great. Thank you to Sarah and David and everyone else for inviting us to do this, allowing us to do this. This is some work that we're pretty excited about, and hopefully you'll find this valuable. Pat, if you could advance. So for the first part of the talk, I'm going to discuss some work that we've been doing on reconstructing the phylogenetic relationships of diatoms. So basically the diatom family tree. And why do we care about this at all? Why do we want to do this? Well, one reason is to understand just sort of like what has given rise to the great species richness of diatoms. So if you look at this tree on the left, you see that diatoms, and I mean, there's all sorts of estimates out there about how many diatom species there are. And it really doesn't matter. There's tens of thousands of species. It doesn't matter to make the point here, but the sister lineage to diatoms has about 20 species. So at some point in the past, they shared a common ancestor and followed very different evolutionary trajectories, where now diatoms have many orders of magnitude more species. And trying to figure that out requires a phylogenetic framework like we're going to be talking about today. The sort of middle graph here shows how we can use phylogenies to understand major ecological transitions. So transitions between marine and fresh waters, for example. So knowing that pattern comes from a phylogeny, and then you can sort of frame mechanistic hypotheses around that. And then finally, the one that's sort of most relevant to today's seminar is that we want to use the phylogeny of diatoms and couple that, use it as the framework for establishing a classification system, because a classification based on a phylogeny has strong predictive power. It allows for very accurate communication and doesn't obscure sort of the underlying biology. So the second part of the talk is going to focus on how we use the diatom phylogeny to then classify diatoms. So if we, most of the work on reconstructing the phylogeny of all diatoms uses DNA sequence data. And this dates back to 1993 when I graduated from high school. Didn't read the paper at that point, but this is a landmark paper by Linda Medlin and Dave Williams and Pat Sims, where they sequenced the 18S ribosomal DNA gene for 11 diatoms. And this is kind of the paper that kick-started the field of molecular phylogenetics of diatoms. Then if you sort of fast forward to 2024, I think the sort of biggest, broadest phylogenetic tree we had for diatoms was published by Teo Nakov back in 2018. And for that paper, he scoured all the sequenced genes available from diatoms. There were 11 genes for about 1,100 taxa. And so that's sort of the progress that we made over 31 years. So the next slide. So if we look at this first column here, the Nakov et al. Column, so he analyzed 11 genes. And diatoms have three genomes: a nuclear genome, they have a genome in the chloroplast, and then they have a mitochondrial genome. So Teo's paper had two genes from the nuclear genome 7 from the plastid genome, and 2 from the mitochondrial genome. The nuclear genome, those are both ribosomal RNA genes, so those are kind of the same gene. And then people can make pretty good arguments that if you've sequenced one plastid gene, adding like 6 or 10 more sort of gives you more of the same signal. And so we have 11 genes out of a possible 10,000. Most diatom genomes have on the order of 10 to 20,000 genes. So we've looked at 2 out of 10 to 20,000, 7 out of a possible 120 genes in the plastid genome, and 2 out of 35 genes in the mitochondrial genome. So Pat, if you put up the next slide. So we have a highly, highly incomplete sample of genomic information. And when you have, just using basic principles of statistics, if you have a very small sample, that sample can be potentially biased. And so it leads to all sorts of questions. So the relationships that over the years have been hard to resolve or uncertain, do those just require more data? Or is it possible that we can't resolve those relationships with more data? Sort of a bad one would be if we're confidently wrong about some relationships. There are some parts of the tree, the diatom tree, that have been resolved with molecular data that don't make a lot of sense. And are we wrong about those? Or are the molecules really right? And so big existential questions. What do we know? What do we not know? And so we're kind of now in this era of phylogenomics. And this term, phylogenomics, has sort of changed in meaning over the years. It was originally coined by Jonathan Eisen as a framework for determining the function of genes using principles of phylogeny. But over the years, it's sort of been co-opted, and now like its common usage is in these endeavors where we use genome scale data, so hundreds or thousands of genes to infer species relationships. And once you have a a big species tree and you have information from hundreds and thousands of genes, then you can ask sort of novel questions about gene and genome evolution. And the really sort of kind of the interesting part of this is that those thousands of genes, by getting a broad perspective on the genome, really reveals sort of the good, the bad, and the really ugly parts of the tree. And the diatom tree has some ugly, interesting parts, as we'll talk about here in a moment. So at the start of this project, there were, if you look on the right there where the total is, there were genome scale data for like 103 diatoms. And most of these were marine planktonic species. And that's because the Moore Foundation funded an initiative to sequence transcriptomes for marine planktonic diatoms. So at the start of this project, there were 103 diatoms that had genome scale data. The vast majority of those were from marine plankton. And so our goal was to fill in these gaps. And so, yeah, Pat, you can keep that. So in our study, we were able to increase the total number of species with genome scale data from 100 to 284. So going from 43 genera, so like almost a factor of 4 or more, up to 175 genera. And you can see we have still a lot of marine plankton, but we've added a lot of freshwater brackish, almost an order of magnitude more benthic species, and several terrestrial species. So that is the data set. And the vast majority of these are transcriptomes, which are all the expressed genes in the cell. And so we combine this with, next slide, we collaborated with Carolina Brilka at Lund University, where for her PhD, she dug into the Cretaceous fossil record of diatoms, and she came up with 17 very well supported fossil calibrations that we could juxtapose onto our tree and use to infer sort of a molecular clock or time scale of diatom evolution. And I'll just talk about like sort of the most surface level of the results here. So a tree is a really good and apt metaphor for a phylogeny because it's a thing that starts and then grows and branches over time. So we're gonna like just sort of walk through in 25 million year intervals the phylogeny of diatoms as we reconstructed it. So this is time here on the left. So we inferred like a time of origin of around 275 million years. And the inference is that ancestrally diatoms were marine, planktonic, and their cell morphology was that they were basically cylinders, what we would call centric diatoms. And then if we advance another 25 million years, more of the same, marine, planktonic, centric, another 25 million years, again, more of the same. And then another 25 million years, again, more of the same. And so what we have here, if you look at this, we have 100 million years of what we would describe as relative stasis in diatom evolution, where they were confined to predominantly to the marine plankton, and their cell morphology was relatively conserved. So if we advance one more, then in the next 25 million year time slice, you can see this tree really begin to expand and grow. And if you can do one more, Pat. So this resolution here, the separation of all these branches, though, sort of belies a lot of uncertainty that exists here. So if you look at these gray bars that we've just put over these nodes, those are confidence intervals in the dates that we estimated for those nodes. And what you'll notice is that those confidence intervals overlap with one another strongly, meaning that we can't say that they're actually significantly different from one another. So what this means, sort of in evolutionary phylogenetic terms, is that this was probably a very rapid radiation that was taking place at this time. And if we sort of fast forward one more, we see that what came out of this rapid diversification down here where there's these gray bars, what emerged from this in this orange and green area of the tree were the polar centric diatoms. And so early on in the evolution of polar centric diatoms, this was a time of major innovation, morphologically, ecologically. It was then that diatoms really became established many different times into fresh waters. There was a rapid diversification of all the major lineages. And what we found, what I'm not going to talk about at length here, is that there was a lot of disagreement among genes. And we looked at like hundreds and in some cases thousands of genes to measure this. And there's a lot of disagreement among genes because this happened so quickly that there wasn't enough time for genes to accumulate changes to capture these events. So as a result, in the polar centric diatoms, there's pretty poor resolution among the major clades. And then if you go one more, what eventually came out of this was the evolution of araphid and then raphid pinnate diatoms. Raphids comprising like the vast majority of diatom diversity, and this being probably one of the most important consequential sort of species radiations in all of eukaryotic microbes. I'm biased, but it's an important radiation. And so next, So we described this, we sort of compared it to a slow burning fuse, where the first 100 million years of diatom evolution was really slow. The rate of change was very slow and highly constrained. And then some 100 million years after their origin, that constraint appears to have been lifted in many different ways, and diatoms from this sort of explosion diversified into the great diversity of forms, morphologically, ecologically, life history innovations that we see today. And so the last slide for me, the upshot of this is that although like most people associate, this is not this group, but most people associate diatoms with the marine plankton, and they've been in the marine plankton the longest. Marine planktonic diatoms are a relatively small fraction of diatom diversity, and most of diatom diversity traces back to that radiation that began in that early polar centric diversification that we reconstructed. And with that, Pat will talk about how we then use this phylogeny to make a new classification of diatoms.
Thanks, Andy. So before we get into the classification, I just want to go through just a little bit of background information about classifications in general and some past attempts at looking at classifications for the diatoms. So, we classify things to create or establish a system of knowledge about relationships. And as Andy said, that knowledge of relationships can open up a lot of doors for us in terms of predicting or interpreting everything from ecology to gene expression or whatever it might be. Having that classification system creates an organizational system for us for the diversity that we have. And of course, it'd be great if the classifications that we had were natural, at least the formal classifications that reflect evolutionary relationships. There are other ways to classify, of course. And while there are other classification systems out there, at least right now, we've decided to go ahead and use a Linnaean system of classification, which aligns with rules of nomenclature and expression and names that are available. We just have to remember sometimes that things get classified together even though they might not look similar to one another. There are cases where overall morphological similarity may not be the best guide to evolutionary relationships. So as I said, we're looking for a natural classification that reflects evolutionary relationships, and there's some examples of those kinds of groups. There are other ways to classify things. We could talk about for identification versus evolutionary relationships are different things. And so we could create classification systems that are convenient, but not necessarily natural. So in approaching this problem, we wanted to leverage the phylogeny that came out of that large study that Andy mentioned and described, and use that to create a classification system. And we decided that, like almost every other group of organisms, we would that are classified, we would use the idea of monophyly as our guide to establishing those groups. Monophyletic groups, of course, are those groups that have the ancestors and all of the descendants of a particular group, as opposed to, say, paraphyletic groups that include an ancestor and some of the descendants. So it was monophyletic groups that we were going to use to identify clades and to identify clades that we would name. So for diatoms, there's been a number of approaches to this. Maybe early on, scientists thought about what might be important, what are important characters to use to then group diatoms in some sort of system. There's a whole history of people thinking of what might be good characters or characters that might be perceived as really complex, and somehow those characters should be used. If they're really complex, they probably didn't evolve more than once, or sometimes the term conservative characters have been used and still with us in the literature that's produced today. There have been times when people made trees. Andy talked about the importance of trees, but maybe how they got that tree might not have been so straightforward or transparent. And even if they have a tree that they created by some analysis of their own, maybe they didn't use monophyly as the approach. And then there were uh, um, publications that created trees. They had formal analysis that they described. A lot of these used molecular data, but they didn't adopt monophyly for the groups that they classified or identified. And finally, for this project, there's trees. There's an explicit formal analysis. We've used molecular data, and we've adopted monophyly as our approach. So just some of these kinds of things. Pfizer had this classification system based on chloroplast size and number. H.L. Smith came up with a group, a three group system that may more or less translate into centric, non-radial centrics and raphids, and then raphids. And then Shute came up with this idea of centrics and pennates. And of course, that centric pennate split was a system that existed for a long time. It integrated valve morphology, chloroplast number, type of sexual reproduction, integrated a number of aspects of diatom biology that people seem to say, yeah, that seems to make a lot of sense. And that system existed for some time. The H.L. Smith system was readopted by Round et al., more or less, in their 1990 publication. Maybe a classification you may not know about, but one by a guy named Bessey, who's thought that the important character was the diatom started out filamentous and became unicellular. One thing about Bessey's system is that the Cerulacea over here off this branch of the Naviculaceae was separated from this branch of the Bacillariales, and that's a feature that we hadn't seen in classifications until molecular data. And of course, Simonsen in '79, his diatom system, he created a tree. And I'm not sure how he got to it, but he created a tree, and then he reflected centrics and pennates, kind of overlaid that. And some of the groups that he identified were monophyletic, but most were not. And then Medley and Kaczmarska, using molecular data, created a tree, and the analysis was explicitly stated, but they identifying these different groups, identifying these different colors, recognized grades of organization, non-monophyletic groups for two of them, rather than clades of relationships. So what we've seen, so this little tree down here, this little cartoon, suggests more or less what a number of different molecular studies have shown in terms of groups of diatoms. And what I want to show here is that from the centric pennate split, if you will, to more modern classifications, none of them have given us a monophyletic, an evolutionary or natural system. So here's Schutz's system. The centrics here are non-monophyletic. The pennates are, based on this analysis. Round et al., the centrics and araphids are non-monophyletic, but the raphids are. Medley and Kaczmarska, radial, non-radial centrics, as well as araphids are non-monophyletic, while the raphids are. Some ways, if you just want to do percentage, we got fewer and fewer percentage of monophyletic groups. And then a system that Dave Mann proposed, and Adel et al. Still had a non-monophyletic radial centric group. So we've never had a classification system that reflected the evolutionary history of the groups, even when we've had phylogenies and formal analysis that showed them. So in the paper that Andy's presentation was based upon, there's a statement in there that said the diatom classification system is broken. And so based on that work, we decided to move forward to utilize the work in that phylogeny that was data rich in terms of number of genes as well as taxa, and to apply that to develop a new classification system. And Matt's going to take us through that, what we did, and take you through a bit of the classification. So Matt?
Thank you, Pat. So yeah, as Pat and Andy already mentioned, we started with this huge phylogenetic phylogenomic data set from the transcriptomes, and sort of use that as a skeleton to look for these clades and identify and name these clades for the classification. So in addition to what was published in the Alverson et al. Paper, which was about 180 genera, we also looked through the literature at molecular phylogenetics over the past 20, 30 years, looking at some of these well-supported clades with just a few multi-gene analyses, right, that seem to persist as more taxa are added and more analyses are run. And so we came up with a series of inferred groups from that. So if we have a clade that has genera in it, which are monophyletic with some of the genera which were already in the phylotranscriptomic data, those were added to our classification, but identified as this inferred group, either by DNA or by morphology. And then the sources of those analyses or the morphology were identified and cited to basically show our work. So, for example, the Thalassius auralis, a very well-studied group of diatoms, both morphologically and molecularly. So any genera that came in the Thalassius auralis in our new classification that had been found in other groups, we put them in there, identified either by DNA, or in the case of the Thalassius auralis, where we do have a morphological synapomorphy that has been proposed in the case of the strutted process, the Fultoportulae, we indicated that such a structure has been identified and cited where that structure came from. All right, so of the 34 orders that had been classified previously, we found a pretty significant portion of those orders were already non-monophyletic and spread across a number of different clades, and in some cases as many as 9 different clades across the trees. And some orders were even across different taxonomic classes in the existing system. So we took the phylogeny, identified sort of the baseline clades, which were the sources of our earliest divergence, our classes. So we ended up with 10 classes, 44 orders, 68 families, and around 440 genera. Most of these groups had already been named, but about 20% of them we had to come up with a new name, a new designation for that group. And yeah, across all classifications, we think this is the one that is the most complete, contains the most genera of any classification that's that's been proposed, and certainly the only one that uses monophyly as the basis for that classification. So our classes, 10 classes, going from bottom or centric to top to the araphid pennates. We've got our Corythrophyceae, our Leptocylindrophyceae, our Melocyrophyceae, our Cocinodiscophyceae, Thalassiocyrophyceae, Bodophiophyceae, Striatellophyceae, Plagiogramophyceae, Fragilariophyceae, and then that old classic, the Bacillariophyceae for the araphid pennates. So in terms of going in here, we're just going to go through a couple of these to just sort of show the process that's in the classification paper. On the right here, you'll see the groups that corresponds to our new classes. On the left-hand side of the tree there, you'll see the classic classification without monophyly. So our first group there, the Corythrophyceae. So in this case, you can see that we've identified the Corythrophyceae down to family and genus with corythrin, which was present in the Alverson paper. But there's also been a couple of fossil corythroids that were well characterized by Kenta Abbey and Rick Jordan's group. So we included that. We included that as inferred families here on the basis of morphology, those flexible, that ring of flexible spines on the edge of the valves for the cells. So we identified precorythrin and microampulla in a inferred family called the Microampullariaceae. And you can see again there, we've included citations for where we pulled this information. So the next class up is Leptocylindrophyceae. So again, these marine planktonic cylinders. So this includes the Leptocylindrales, the Leptocylindraceae, and then of course the Leptocylindrus and Tenuicylindrus from the Ninjapa and Zingon paper that really explored the diversity of that group years ago. And then we get into some of the more diverse groups, including the Melocyrophyceae, this new class. So this includes the subclass Proboscidophyceae, sorry about that, the Proboscideales, and the Proboscideaceae, which is proposed by Nikolaev and Harwood. And again, we've got some fossil taxa here that are seem to have a lot in common morphologically than the Proboscia that was included in the phylogenomic analysis. So again, we've included those as inferred genera on the basis of morphology with citations. And then we get into the other subclass there, the Melocyrophyceae, with the Stephanopixidales, Stephanopixidacea, Stephanopixis, which was included in the analysis, and then genera inferred from the morphology. These mostly fossil genera here. I mean, nope, entirely fossil genera there with Costopixis, Creswellia, Pixilla, and Cystophenia. And then to round out the Melocyrophyceae, we have our Melocyrales, which again, for the phylogenetic analysis, the phylotranscriptomic analysis included Melocyra and Alicocyra, and then we included Alveophora based on the morphology published in Ulotseva and Titova 2019. Okay, well, and one more there with the Paraliales. Sorry, forgot about the order Paraliales. Paraliaceae, again, an existing family. Paralia and Dictia Podocyra were included in the phylotranscriptomic analyses, and then Bipola was inferred on the basis of overall morphological similarity by Nikolaev and Harwood. Okay. And I think Pat?
Yeah, I'll take it from here. Thanks. So we're well aware that any new classification system has challenges, and we're happy with all the data that it's based upon, and it's what must have to be the most transparent system. People should be able to, as they go through all 10 of those clades, see what what we looked at as the data to support the system that we came up with. But there's always challenges to these systems, and one of them is that the basis for the backbone, as Matt mentioned, was from the system that Andy and his colleagues put together for the PNAS paper, and we don't have synapomorphies that would diagnose each of those lineages. So that's a shortcoming, and that's kind of related to another thing for diatomists, is that we actually don't have an overall morphological analysis for the relationships of diatoms. We could map features onto this tree, but we don't have, and those features could be sexual reproduction or symmetry or whether there's a raphy present, but we don't have an independent tree related to morphology, based on morphology. And a good question for diatomists would be why? Why don't we have that? And because of we don't have a tree based on morphology, and we can't use exclusively fossil taxa to contribute to the development of the tree, we don't know about the unique roles that fossils might play in the phylogenetic reconstruction of diatoms. For higher plants, for a lot of different vertebrate groups, fossils have been shown to help give answers that might not have come from molecular data. And so based on this current system, we don't have that incorporated into the system. So those three things are kind of related to each other. And then we also have this system. When you read some of the descriptions, they're pretty generalized for the new taxa. And so trying to write a description that has a variety of morphological entities in it, and you end up writing sometimes descriptions that are very general. And another topic that might be for discussion here is whether if you come up with a description with diagnostic characters, does everything for a group, does everything in the group need to have those diagnostic characters? The paper for the classification has just been accepted at Journal of Ecology. This QR code, and I want to thank Amy Chang for putting this together for us. You can download the current version of it. It hasn't been published yet, but if you point your cell phones to that, you should be able to get to the paper and have a look, and you can see how that whole classification system works out for all 10 clades. Matt just went through the first 3 just to give you a sense of how we did it. If we were to present all 10 clades, we would have scheduled the Diatom Web Academy for the day rather than for the hour. And the next steps, we will incorporate this system into Diatom Base. We will develop an Excel file that basically has this classification and offer it to AlgaeBase. And we hope that Mike Gyrey will embrace that. But I don't know if you ever looked at the classification system in AlgaeBase, but for diatoms. So here in the phylum, the heteroconts, there are 3 subphylums, 2 of them related to diatoms, but there's not one group for diatoms. So in this classification system, there's not even a single monophyletic group called diatoms in the AlgaeBase classification system. But anyway, we will offer this up if Mike is interested in it. Diatomists generally, not just the 3 of us who have participated in this, but there's a lot of work to do still. And I want to send a message that this classification system is a jumping off point, not an ending point for the work that folks have to do. I've already mentioned the need for a morphological phylogeny of diatoms, but we still have these groupings of people using good features, conservative features, complex structures to group diatoms. And by using a phylogenetic approach, accepting monophyly as the way to identify groups, I think there's a lot of great work still to come for diatom phylogenomics and developing the natural classification system for this group. So we appreciate your attention, and we're looking forward to your questions. You are welcome to address them to all of us or any of us. So thanks a lot, and we look forward to your questions.
Thank you, Pat and Andy and Matt. And if anyone has a question, you can raise your virtual hand, or you can put your question in the chat, or you can just turn on your camera and let us know that you have a question.
Just shout it out. Just shout it out.
I'll go first. I'll jump in there. And hopefully my connection is stable enough. Does this, I'm looking at this work almost like I feel like we work on different planets and how we look at diatoms, not saying that we're in disagreement, but just the tools you use versus what I use. Does this sort of analysis lend itself to understand the variability we see in barcode regions and how well those barcodes can be used to characterize diatoms as a whole or certain groups, like looking at does 18S resolve certain areas as well as RBCL or not?
I'll try that. It does and it doesn't. We have some work here. So like barcoding often uses like a fragment of the 18S. So if you look like the Terra Oceans, they've generated like a bajillion 18S sequences for diatoms. And those do a good job at some level, but you can only expect so much from, 100 nucleotides in a genome. And we have a project right now, for example, where it's on this tiny little Thalassocyra profunda, this tiny little diatom. And the barcodes say that everything's identical, but we've sequenced whole genomes, and there's like 4 sort of hidden species in there when you look at the level of the whole genome. So does this study, help you in any way with barcoding? No, but knowing that like there's probably there could very well be more there in terms of like diversity than a barcode can show you is good to know. I think it's good to know.
Yep. Thanks, Eddie. I want to just pop in here and remind us what barcodes are for. They're for distinguishing species, right? The ideal barcode sequence has an individual base pair for every single species, right? When we're trying to do these phylogenetic studies, we're looking for shared characters, not unique characters, right? So the barcode regions might be great for identifying species. They're really sort of not great at all for building these phylogenies and classifications. So kind of apples and oranges there, and what we're trying to do.
Okay, question from Anna in the chat. Anna says, a big reclassification of diatoms feels a little premature. How many genes have you used? I glanced at the method sections and saw 11, which would be amazing 20 years ago. Now papers for flowering plants routinely use a couple hundred for even trivial questions. How do you feel about limiting sequencing of diatoms?
So the tree that we based this classification on, we published earlier this year, and we used anywhere from, I think, like 200 to 2400 genes. So we did like a whole array of analyses and looked at like different methods and different size data sets. So if you look at like the recent, so she mentions flowering plants, like the most recent phylogenomic study of global study of flowering plants used 353 genes. And so we're definitely like on par with what they did. So I think that we use far more than 11, and I think that given that I don't think it's premature with the tree that we have to reclassify, but people could disagree. But there might be
I
a
have kind of
little
a follow-up
yeah.
question to that. Maybe, I mean, I just know next to
Yeah.
nothing about sequencing, but transcriptomes sound like they have some, if they're things that are expressed, they have some variability. Do you have a way to, sort of poke at that? Say, if there's a certain expression, like you grow something in a, I don't know, in a, in a high silica concentration, and then you grow it a low, and you kind of see like how much does that change?
Yeah.
Genes are expressed.
Yeah. So for this study that we talked about, we don't have like the replication necessary to do that, but we've done that for in an experimental setting where you have like proper replication and everything so that you can say, the expression of this gene is significantly different in this condition versus that. And we've done that a lot in as you change salinity to see how diatoms have sort of gotten into fresh water and acclimate to fresh water. But you can absolutely do that with transcriptomes. But for this study, we just sequenced one, and you don't really have like statistical power to be able to say anything. So we just use like the data in the genes themselves.
Okay, Layla has her yellow hand up. Layla, would you like to ask your question?
Yes, thank you. I wanted just to ask you if you can give an example how Melosira and Proboscia are the same in the same plate that you showed us, Matt. I mean, Proboscia was before Rhizosolenia. It seems completely different. How you put it together. And my second question, we use it to use the names of species and as they were described, and after see the statue of name in alga vase. So now what we should do, and are we going from always from centric to araphid and araphid in terms of evolution? Thank you very much.
Well, I guess I'll take a stab at that. Certainly with the association with Proboscis and Melosira. In terms of the two genera, yeah, you're right. There's not a heck of a lot there in terms of the similarity, other than broad categories like the presence of labiate processes and loculate valve structure. But there's still a significant amount of difference between those. I mean, those share a class, right? So within that class, they are within two different subclasses. So yes, despite the fact that the molecules put them more closely related to each other than to other groups of diatoms, there's still a lot of diversity in there and a lot of nodes between them. So does that answer your question at all?
Yes, thank you. The second question, it was just when we use it to take from Algabase the statute of name when the name changes. Now with your classification, your new classification, how we would do, since you said that maybe with Algabase it wouldn't work.
Well, so I'll try my hand at that. So if you're talking about higher level classification, you know,
Yes.
we'd expect that. So what we could ask Mike, what is he thinking about with his classification? Where did that come from? I can't even tell you what classification is used in AlgaBase based on the fact that they don't even have it as diatoms as a single group. They have diatoms in two separate groups. So, so it's a fair question to ask, what classification is AlgaBase using? What's that based on? So at least from this, you will be able to say, there's the phylogeny, here's what they did, here's the basis for their decision. So we hope that he'll adopt this phylogenetic classification, but in at any level of utilizing names in AlgaBase, you should ask yourself, how is that the accepted name? And it's not always clear.
Okay, and for you, the evolution from centric to araphid, still the same way that we always said. I mean, araphid and biraphid.
Andy, this is your question, of course, but I'll just say the course of evolution that Andy described would still have that general progression from those groups of convenience that you mentioned, from the earliest to the most recent divergences.
Thank you very much.
Hey, Mihaela has a question. I would like to ask about your vision on the possible impacts of the new classification on practical applications that we are now using based on diatom morphology for paleo and ecological applications.
Well, I'll repeat something that Andy said previously, and we would expect with this new classification to, if you believe that there are trends or patterns in your data set that are related at a higher level of, so this more or less, we're not changing any genera. We haven't checked, we haven't done anything at that level. So if you think you're seeing ecological or paleoecological patterns at the level of family, order, those kinds of things, we believe that there'll be less noise in this system than if you were using Medlin and Kazmarska or Adel et al. So if those patterns exist at that level of hierarchy, we'd say that this ought to be help you interpret those data rather than previous classification systems. So we haven't touched. I think the paper actually has one genus described in it, but we haven't really touched the genus level classification. We've just tried to assemble those genera into a system that reflect their monophyletic groups.
Okay, David says, darn. I hoped we're going to get a resolution to Lindavia / Pantoxiella. Tom.
Good afternoon. Yeah, I had a question going back to the file. You're going back in time. So you saw this diversification about 150 million years ago. What was going on at the planet? Maybe with the the breakup of Pangea and the Cretaceous, and comment on how that may have produced the, what we're seeing, where we see a dominance of diversity in the freshwater and dominance in benthic species.
Yeah. I wish that Jeffrey was here. Is he here? Jeffrey Stone. He emailed me within 2 to 3 minutes of our paper being published, because we didn't really try to layer on sort of environmental Earth history hypothesis. And I'm always kind of hesitant to do that, because it does feel a bit like storytelling to me. But Jeffrey pointed out, what is it called? The Western Interior Seaway or something opened up at around this time and would have created lots of new sort of benthic habitats. And so, yeah, you could say that. I mean, it's definitely something that would be consistent, but I'm not super comfortable. Yeah. I mean, there's a lot of speculation that you could do, but whether that is related to what happened in diatoms or not clear.
I do want to, let me just say that it's not going to answer your question, Tom, but I think that one thing that maybe Andy downplayed here that should be stated is that this interpretation of diatom evolution is pretty much entirely the opposite of what most people have said the last 30 to 40 years. So if you go back to those papers by Round and Crawford or lots of other people, they envisioned rapid initial diversification of diatoms in a habitat that was where most of the things were attached. So they would have ocelli or some other features that would create this kind of coastal environment. These data don't support any of those hypotheses. In fact, it's a marine plankton slow fuse for a long time. And that's a really different interpretation for what diatoms were doing at the early stages of their evolutionary history.
I'd jump in with a follow-up on that to say that it's quite remarkable that in that slow fuse period that you're passing the Permian extinction and late Triassic without any major change in the evolution, which is pretty phenomenal. But I did have a question and wanted to ask, you mentioned trying to link these genetic data to be able to use in interpreting and informing with the fossil record. What sort of database or like traits would you start to look at in heading in that direction? And who wants to write that grant with me?
Well, I will absolutely write that grant with you. I've got a couple of partial proposals written down sitting on my hard drive. But there have been a couple attempts to sort of marry the two. I know Andy already mentioned Carolina Brilka, who did her Cretaceous diatom database, which proposed those what was it, 17, 18 calibration points. So the idea is generally to hope that there are some features that maybe haven't been examined fully by electron microscopy that we might be able to link them together. But also the diatoms have been affected by mass extinctions. You know, Andy mentioned the lack of resolution and the disagreement in the polar centrics, and there's been a lot of extinction in that group. So that's the diatoms have certainly been affected by some of those events, I think.
But on the other hand, not affected by the KT at the KT boundary. I mean, they just go past the KT boundary with vigor. And you know, what exactly was happening there and how that happened when, 90% of the rest of the Earth's biota was wiped out. is a super interesting question.
Lots of super interesting questions, and we're past our hour. So I'd like to thank Andy and Pat and Matt for your work, and thank you for bringing it, sharing it with all of us today. And I'm sure you will hear questions from people in the audience. This really covers the huge range of diatom work. So it's really a big deal, and very much appreciate what you've done and sharing it with us today.
Thanks,
for
Sarah.
us. Yep. Thanks, everyone.
Yeah,
Take
take
care.
care.